Okay, welcome to BoneChat discussion 132. Today we're joined by Silver Bullet Therapeutics co-founder CEO Paul Chirico to discuss their new low-cost micro-galvanic coating technology that is going to disrupt orthopedics because it offers a zero-cost clinical upgrade to prevent surgical site infections. I mean, this is brilliant. I'm turning the mic over to Paul and welcome aboard.
Thank you. Thank you very much. I am just going to go right into the presentation.
Got
Let's
it.
see if I
Perfect.
can move that back. All right. Everybody can see the slides.
Yes.
I'm Paul Chirico, President CEO. I'm also one of the founders. I have been in the industry for, oh gosh, now this is my 47th year, 48th year.
Wow.
So I'm starting to add up. Cut my teeth in orthopedics and spine at Esculap. So I take a lot of pride in spending time with Esculap, and Tiger and I have run across each other. I was the previous founder of a company called Spine Align Medical. You may remember some of that, and that was a nickel titanium insert that went into the vertebral body to create a kyphoplasty or vertebroplasty type approach. So with that said, um, I'm taking the approach today. I want to thank Tiger for inviting us to present. But I'm going to take his approach to presenting, and that is a little bit more educational. Rather than just a self-serving presentation, I really wanted to present it from the perspective of what have been the challenges of going to market and developing a technology such as this. There really is no competitors out there. I mean, there are certain people dancing around the topic, and there have been a lot of talk about it. But, at the end of the day, is there been a commercial competitor or a commercial success out there? And there really hasn't been. It's not for lack of need. I mean, this was a report from the CDC, and I tried to pick up something fresh from just January. And you can just see that surgical site infections, or SSIs, as well as hospital-acquired infections, account for a tremendous amount of cost and to the system overall. And that's not just a cost to the system in financial dollars, but as you can see there, it actually shows that there's a huge increase in mortality. And this is even post recovery. So it's almost like when a person is exposed to an infection, hip infections are a good example. There was a study years ago that patients who actually had a hip infection had a mortality rate that was greater than patients who were recovering cancer survivors. So it predisposes you in this area. So this is a significant area that needs a lot of attention. Big costs. Certainly, we have the rise of superbugs and that are occurring worldwide. And even if they're as far away as Sri Lanka, they're only a plane trip away from being in the United States. So with a world economy and a world travel, as we found with COVID, it doesn't take long for something to move quickly. So there are challenges to developing a technology, an antimicrobial coating specifically, and I'm going to address these. The coating needs to deliver antimicrobial activity immediately. You can't have something that builds up over time and then has an antimicrobial effect, or in the world of bacteria, meets the minimal inhibitory concentration to kill the bacteria 5 to 7 days from now. At that time, either the patient's going to be dead or you're going to have a huge problem associated with removing hardware and then treating the infection. The other thing is, our company is funded primarily with clinicians. And the one thing that they have told me all the time, don't make it a square dance, Paul. It has to be something that can fit into how medicine is practiced today or surgically is practiced today. So you don't want to make it something that it requires sitting in a bowl of saline for 20 minutes to pre-soaked, and then you have to use it within 2 minutes. It has to fit into how surgery is done. The third one is it can't be toxic to mammalian cells. So everybody says, well, when I put a screw in, it's only going to have a certain amount of antimicrobials attached to it. Actually, it's not. You have to start thinking in terms of, and this is what's wonderful about the FDA, is that they're pretty smart people there. So they say, hey, wait a minute, Paul, you can't just say the silver bone screw is going to have this amount of silver. In the case of a bilateral ankle fracture, you may use two plates, two 12-hole plates, which have now 12 3.5 millimeter screws times two, and then two big 4.5 millimeter screws running across the fracture plane. So no longer are you thinking of just the antimicrobial components of just one screw, it's 26 screws. And so you have to start thinking in terms of that. Regulatory clearance is critical as well. And I'll talk about that because in today's world, being a 510K means more than a lot of people think, especially when it comes to established reimbursements or DRGs, and then fitting into the system that was already existing, rather than trying to go off and getting it through the regulatory process, and now you have to go through the reimbursement process. And last is, patents are the critical element here of making it fundable, to making sure that you have the capability and the legs to be able to support this, and whoever comes behind you and helps you fund this company is protected. So with that said, I also talk about the specific needs clinically. This is what I call my hierarchy of complications. At the lowest level of an infection, you're just going to spend a couple more days in the hospital. You'll take some antibiotics, and then you're out. And believe it or not, the cost on that can also be significant. And that just a patient that'll cost you another $6,000. But as you move up the hierarchy of complications, as the infection becomes more critical, you end up with malunions, with a potential for permanent disability, ultimately an amputation, and then potentially death. I always tell the story here. I had a friend of mine, 60-year-old soccer friend, soccer buddy. He has been my friend since first grade, but he was the varsity women's coach at the local high school. And the secret of his success was he ran up and down the field with his players so he could coach them. He was on a camping trip, tripped on a rock, broke his ankle, and actually when he came back, they put the hardware in, it became infected, and now he has a malunion. And unfortunately, he can no longer run up and down that field. So he had to give up his love of coaching women's soccer because of that. And so it happens all the time. Okay, most bacteria at the end of the day, they're doubling every 20 to 30 minutes. And we did some tests on this where we actually took silver-plated screws, just silver-plated screws, and put them into a medium and exposed them to MRSA. And as you can see, that slide down below is where the silver just doesn't release quick enough. The silver ions are what kills the bacteria, just doesn't release quick enough. The bacteria grow right up and over the top of it, and it behaves just like a control. So you have to develop something that's different. And in our case, we have developed a blend of silver and platinum where we have two dissimilar metals. And actually, you can see in the lower graph is that at 24 hours, we've got a not 100% kill, but we've got a kill ratio of where we can no longer detect bacteria. And we can actually show that on the auger slide above, where you can see the auger completely is cleared out by the silver ions that are now that are releasing into the auger, as opposed to a screw with no protection. Again, bacteria grows right up and over the top. And it has to work on a variety of bacteria. This is quite an R chart, and I apologize for it, but what the take-home message here is, these were the superbugs that we tested our product against. E. coli, Pseudomonas, they have to work against gram-negative and gram-positive. And in this case, we had, in some of the slides were the blue, and in some of the slides were actually the green line. But all of them showed that we had a kill ratio that killed all the bacteria at 24 hours. And these were the superbugs. So this was a study that we actually commissioned outside of our company. And then we actually published this as a press release because we thought it was so important. Here's how our technology works. All we do is we add silver in the presence of platinum, and we've developed, it took years to develop the blend where they're in the same plane. And why are they in the same plane? Because traditionally, when they've developed galvanic type systems, you end up having a plane of platinum or palladium, and then you put silver or zinc on top of it. But what you've done is you've isolated the component which causes the silver to corrode off or ionize off. And as you can see here, we've got a release rate for what we call antibacterian, and we release it in 24 hours. So we get a peak at 24 hours, and it tails off over time until it's completely covered with new bone, or it's subtherapeutic and it just releases a very small amount. So you can see we address developing address killing the bacteria when you need it the most. Most technologies are either an antibiotic, or where they're preloaded with an antibiotic that is not resistant to abrasion, especially in orthopedics. It tends to be highly toxic to mammalian cells, because now you're delivering so much of it, because it's pretty much gone at 6 or 7 days. So this is what we've developed, is a feedback mechanism that looks like a time release, but basically they're metals. And so this is the secret of our technology.
So I'm no scientist. So what is OH? So you basically emit silver ions and OH. What is?
That's right. So what we do is, in the presence of water and oxygen, platinum acts as a catalyst and takes silver and steals an electron away from it, breaks down the water to OH negative, and then spins off a silver ion, which is missing an electron. So and the platinum is untouched because it's a catalyst, and it's probably the safest metal in the body that there is. So it's the body tolerates platinum quite well. So as a result, we deplete the silver ions over time, and around the silver by stealing an ion. And then what happens to the ion? It gets picked up by a white blood cell, and then it's disposed of through the liver. And it's got to work against biofilms too, not just active bacteria, but biofilms. This was a study that was commissioned by Straumann dental implants, and a Dr. Zipman, who is in Switzerland, and they actually compared several different technologies and against biofilm. And you can see down below that they've used everything from um, um,
Zirconia.
zirconium to titanium to there's a whole host of different
Yeah.
different types of and I can provide that paper to you. And what they found was the AGPT, our blend reduces biofilm significantly. And so not only do we kill active bacteria, but we also work on biofilm as well. And this was a peer-reviewed study that was published a few years ago. So now what we have is a technology that works on bacteria and it works on multiple bacteria and it works quickly. But again, what I mentioned before is it has to be a technology that fits into the day-to-day current ways of surgical practice. Believe it or not, surgery in Europe is done a little bit different than surgery, or especially orthopedics, in the United States. In Europe, they tend to go and take out their plates and screws almost within 9 to 10 weeks. In the United States, we tend to leave our plates and screws in for a long period of time and oftentimes permanently. So our product in this case is approved in Europe. And as you can see, we needed an indication for use that didn't change how you did things. Now, what this is it basically says if you've had a prior infection or you're a high risk patient, and patients who are high risk, I think all of us now over the age of 45 are considered high risk patients. Anybody with a comorbidity of smoking or alcohol use is a high risk patient. So, what we have now is a what we call our orthofusion bone screw system that can be used for open reduction and internal fixation, indicated for high risk patients. That's a major breakthrough for certainly the EU. And we're still working with the FDA here, and we anticipate that we'll have the same indication here in the United States one day. But it also has to fit, as I mentioned before, the common practice of contouring your bone plates. Then you can see here, we even have marks. You can detect a few of the marks here, right here, where we actually use the pliers to contour or bend the plates at extreme conditions to see if we would have any delamination of the coating or flaking. And as you can see, we don't have any. So this, again, orthopedic surgeons are known to not necessarily be the kindest on using on their tools and their plates, and for good reason. And and we have developed a coating, so the coating can't be susceptible to rubbing off or abrasive surfaces or bending or contouring. It has to be durable. And so that is a critical element as well. The other thing is it has to fit a lot of product. That's a big problem. And as you can see here, this is just a listing of the bone screws that we have approved in Europe, and it's in the 4 to 5,000 pattern range. So it's a lot of product that you actually have to have that kind of fits into everyday need. And there's always that one surgeon that needs that headless 1.8 millimeter screw, and you're not going to have it. So we try our best, but the numbers still are pretty big. And again, you can't change the methodology of how surgeons use the product. You have to fit their needs. Fitting their needs in other areas as well. Dental implants. We have been able to effectively coat both pacemakers and defibrillators with intact electronics, where they actually took the pacemakers and defibrillators off the quality line, shipped them to us, had us coat them, and then shipped them back to the company to do all their water bath testing as well as antimicrobial testing. And as you can see, there's ways of coating. Defibrillators are part of the electrical circuit. So you actually, when it charges and sends a charge to the heart, it actually comes back to the can to complete the circuit. So if you fully coated the can, then you could potentially cause it to be insulated. So we had to come up with novel ways of striping, and still we had great antimicrobial activity, and yet we didn't have any effect on the operations of both pacemakers and defibrillators. And we can also coat mesh as well as, like I said, dental implants. So again, fitting the need is critical because again, there is a technology out there for pacemakers and defibrillators which consists of a mesh envelope. Well, when you think about it, not only now it adds another $1,000 or $1,500 to the procedure, but the cardiologist has to figure out how to put the mesh, the defibrillator or pacemaker into the mesh bag, make sure the leads are not interfered with, and then tuck it into the pocket that you've just made while the mesh is starting to get mushy, and because it's subjected to fluids of the body. So in this case, you could just slip it right in. Well, now we are on to impurities and byproduct. And again, what this is our product, for instance, when we coat our product with silver, it is 99.997% pure. The platinum is 99.997% pure. Why? Because there were companies over the last 20 years that were producing silver coated products that were 97% pure. Well, suddenly what's in that 3% and how much does that add up? And as you can see here, our impurities, and every product has some type of impurity, is measured in the parts per hundreds of billions. So and again, you have to sum the number of products you have up. So if you have 24 screws, it's 24 times the impurity, not one. And so that adds the plates, screws, etc. And so in this particular case, what the byproducts can be toxic to the body or the tissue, surrounding tissue. And this was important, and we ran these studies. There were, a lot of, when you start thinking about the amount of science that goes into developing a coating such as this, it goes on it takes several years to accumulate this amount of data. And it's critical because, like I mentioned before, I have the utmost respect for the FDA because if you don't have it, they're going to find out that you don't have it. And so you can't hide. This also includes all your biocompatibility testing. You have to do your cytotoxic testing, your irritation, sensitivity. Certainly, there have been other technologies on the market that claim that they are nanotechnologies, nano silver, nano zinc. Nano just means that where we're operating at the elemental level, a silver element, one atom, Nano is just a big chunk of something. So it's a larger piece, but it's still at the nano level. Nano products still are genotoxic. And so rarely do these pass genotoxicity tests. That's critical because as you go through the EU process and as you go through the FDA process, they're well aware of that. So getting a product cleared or approved that is genotoxic is never going to happen. So again, um, it's that is another reason why more technologies have not been developed in this area because of the limitations of the toxicity. Now, we all know that the best way to protect a new implant or an orthopedic implant for against infection or any other thing is to add new tissue to it rapidly. And so technologies can't inhibit or retard new bone being laid down. And again, this is a study that's going to be published soon. Again, another study, a dental implant published by it was funded and driven by Straumann. And as you can see here, in these lower slides, we actually triggered a rapid tissue increase. So the first question is, why? How did that happen? Again, I'd mentioned before, we're putting two dissimilar metals and we're causing an electronic galvanic system. So we're generating a very, very small current. And what's a small current do? It actually is a tissue stimulator. And we're seeing that result in these slides here and in these electron micrographs, as well as the laser microscopy that was done to show that we had significant new tissue growth onto our implants. So again, looking at developing a technology, all these start adding up to why hasn't this market been captured before, or why hasn't another technology been on it? Because there are a lot of challenges to getting a product scientifically that will not only kill bacteria, but not kill the patient as well. 510(k) process. All of us are probably experienced people that know that in today's world, getting a new product that's expensive across the hurdle and into a hospital is going to be pretty oftentimes ignored or pretty limited. And so PMAs and de novos, which require, a lot of work, but not only that, it triggers a new classification and oftentimes a new reimbursement. So you fought yourself through the regulatory process. You're now on the market, you're commercial, you're ready to hand it over to your sales organization, and they realize that they have to go hospital system by hospital system and fight for reimbursement. So that slows adoption significantly. 510(k)s then are still a benefit because they have established classifications and you fit under a classification, in our case, a bone screw, and we then can fit existing DRG. So as a result, if your product is not extremely expensive, it fits right in and it has established reimbursement and as a result. So again, a major hurdle, a major challenge to getting on the market. Drug-based products, which, are combination products, which involve, drugs, I don't even know how those reimbursement methods work anymore. So how or who
that's a
you
red
go
hole.
to?
Yeah.
Do you go to the PBM? Do you go to the hospital? Who pays for that? And again, on this another challenge of getting a technology. So the silver bullet technology we are a 510(k) product, and we fit established classifications and reimburse DRGs. So we fit right into the system. And again, we don't force the surgeon to change the way they do things.
That's huge, Paul. So you're basically a 510K with some clinical work is
We're a
what they're
510(k)
giving.
with some clinical work.
Got it.
Patents, I can't speak enough about it. When we first started the company, I remember some of my VC friends would look at me and go, Paul, give me a break. It's silver. It's been around for a couple hundred years. You'll never get a patent. Give me a no, it'll not happen. Well, it's not just silver. We've developed a silver releasing system. We had developed a system by which we can use same sputtering in the same plane, and we interlace the silver and platinum together. So when they are exposed to oxygen and fluid or water, in case body saline, they're in the same plane. So they create the galvanic cell and reaction as a result. So we're north of 30 patents now, and I think we have almost the same number in Europe. In fact, at one point in time, several of our investors wanted us to silo the company and create 5 separate companies: an orthopedics company, where the IP was supportive of that company, a dental implant company, a surgical company, which was surgical mesh and suture, a cardiovascular company, and I'm a simpleton. I can't. It boggles my mind just keeping track of everything in one company, let alone 6 or 7.
Yeah.
So,
That's complexity. That's what Osteocentric went through. They tried to license to every vertical. It's really hard to do.
yeah.
Um, yeah,
And you know something, Tiger, at the end of the day, when you're licensing it to somebody and they feel that they're missing out, they feel like you're cheating them by saying, okay, you can't have the cardiovascular.
yeah. You just have
So
this little piece right here.
this piece here. So
Yeah,
it's better to go through the negotiation phase and then find out that it falls out, and then you can take the cardiovascular and move it into something else. So that's my experience.
So
And
so you have a huge patent waterfront, and you're not patenting silver implants. You're patenting processes and methods. I assume.
that's right, as well as
Yeah,
know-how. I mean,
yeah,
not everything gets captured in patents anymore. There is a certain amount of know-how, and that is critical to, I think know-how is in some respects growing as even more important than patents in the
yeah.
future.
Trade. So you didn't go trade secret. A lot of people go trade secret if they don't want to tell the world what they're doing, but um, there's pros and cons, obviously. But
Uh, somebody could try to reverse engineer our product and they were not going to get the same performance.
yeah, no, I get it.
And that's one of the things that we're proud of as a company. So in summary, uh, when coatings are embedded in polymers or another material, the polymer tends to rely on breaking down or to release the antimicrobial. And this affects the effectiveness, it affects the timing, and it affects the tissue response. So, so if you have something built into zeolite or a polymer that wears down, it's not as predictable in how it's going to break down in my body versus somebody else's body, or different temperature, or so in our case we are exposed to fluid and oxygen, and as a result, the silver ionization that occurs, the feedback mechanism is once the ions get to a certain level, it shuts off. And then when it drops below that, it turns back on. So this is what keeps it kind of as a time release over time. Again, most coatings relying on like rifampin or minocycline or something else, when you're adding up, again, you have to think of the sum of the number of devices that you're putting in especially orthopedics. It's not just one mesh area. It's 24 or 36 screws. This can actually increase the toxicity, and it can also increase the run out. So, you're trying to adjust for the run out that's occurring in just 7 days. So you increase the amount, and what you're doing is now you're increasing the amount, so it's toxic. Again, most coatings, especially in orthopedics, they have to be handled with kid gloves. I remember reading the IFU for one company that had developed a silver device, and it said, when you remove it from the packaging, use gloves because the coating is so unstable
Fragile.
and
Wow.
fragile. So, I
Wow.
mean, um, and you cannot contour it. So, like in the case of a plate, you couldn't take it out and take a pliers and adjust it for the anatomy of the patient that's required. Some drugs, coatings are drugs or PMAs, and they're just going to be too expensive to adopt long term. We have developed our product where we add basically about 5% to the cost of goods. And in the case of, you take a bone screw, well, titanium bone screw is an example. Most 4 millimeter by 10 millimeter length titanium bone screws have a list price of around $350. Now, no hospital and nobody in their right mind pays $350. It's around $100. So it's the list price versus the actual ASP is about $100.
Okay.
What do they make the bone screw for? They buy them or they make them for around, we get them for around $6 a piece. By the time we coat them and sterilize them and package them, we're at, say, $10. We're
Wow.
still in the 90% profit margin.
Yeah, that's really cheap, and that talks about the volume of silver and platinum that you're using. You're using, since it's angstroms thick, you're just barely using any silver or platinum.
That's right,
That's
just enough. That's the
just
way we
incredible.
describe it. We use just enough.
Yeah.
So in our case, our coatings not only um, some coatings retard new tissue attachment, new bone growth. In our case, we actually have studies now that show that we actually increase tissue attachment. And there are some new technologies out there that are not coatings at all. So I know of one technology where they have a knee implant, and if it gets biofilm, now you have to put a needle down and touch the implant, and basically it heats the implant up. Well, you've just introduced a new source of bacteria because now
Hmm.
you've got an invasive procedure going in. You have to evacuate the bacteria off that is now in the synovial fluid. So now you have to remove that because that's a potential source of, I mean, they could form biofilm and then show up somewhere else. So
And not to mention, it's a second surgery, and there's a whole team and an OR needed. Yeah.
there you go. And that's an example of something that changes the way we're supposed to do. What we'll ultimately be able to do is work with, and we're not going to have a knee. We're not going to develop our own knee. We're not going to develop our own hip or our own shoulders. But we would like to partner off with somebody who already has their knee, hip, shoulder, that we can actually show that we can coat all the non-articulating surfaces. You don't want to coat the articulating surface because that's
No.
designed to be smooth and it's supposed to articulate a certain way. So you don't want to introduce something like cobalt chromium into this area. You want to be able to go into the area that touches bone. So at some point in time, we would look for a partner in those areas because we're not going to develop it, just like dental
Right.
implants. We're not going to develop our own dental implant, but there are enough big companies out there that will need something like this.
Yeah. Paul, let's turn off the slides and open it up
Yeah, with
for
that said,
everybody. Yeah, I'm going to try to. So who's got the first question? Try to bring everybody in. Go ahead, Todd. You're up first.
Paul, super interesting stuff. I like it. Couple questions. You showed a stainless steel trauma plate. Do you have material restrictions? Can you coat titanium, CP tie?
When we first developed the company, long-winded answer. We never intended it to be a platform technology. We always thought of it as a technology that would coat orthopedic implants. We never dreamed that we could be able to coat PEEK, we can coat mesh, we can coat stainless steel, titanium. The only material that we have not been able to get it to stick to is PTFE. And you know something? We'll give up that one.
Who cares? Yeah.
Awesome.
Good
Thanks.
question. Yeah, I'm kind of interested in the European experience. Can you tell the group what was involved in getting the CE mark, other than just testing, and then talk about pricing a little bit in Europe, silver bullet screws versus regular screws.
So we're taking a more aggressive approach in Europe, and that is one of the companies we're working with. You may know him, and we hope that everything moves forward with him. It's a company called Intumed, Adrian Ford. We've been talking about the public health system in the UK as an example, and in the UK, getting new technologies in, especially when you have dominant companies that control the tenders there. So, you have a multi-year tender for plates and screws. How are you going to get your product in on the market? Well, we're not just a standard titanium plate and screw company. We're actually an antimicrobial. So in this particular case, they can walk in and say, we have an antimicrobial line, and they'll say, well, what's the price? The pricing is going to be, we can price our product at parity to the market without losing and actually gain market share. So we're not going to go out there and be the company that says, hey, we have a new mousetrap, we can get 25% bump in the price.
Well, that just takes the price argument right off the table.
We're gonna throw all of our MBA experience of pricing out the window, and we're gonna go in and we're gonna price at parity, because we'll have a reduced margin, but we'll gain market share quicker.
Yeah, I like it. And then tell us about the 120 patient trial in the US. What is it? When did it start? And what are the
we
endpoints?
haven't started it yet.
Okay.
And the reason for it is we thought we had a partner on that one. And the reason for it is it's a 120 patient trial. And in some respects, I dragged my feet on it because we wanted to start it. It's only ankle fractures. It's lateral ankle fractures.
Open only, or open, right?
All open.
Okay.
Yep. So they're all ORIF procedures, and they can use anywhere between a 7-hole and a 12-hole plate. So traditionally, you would use 3.5 millimeter by 20 to 24 millimeter screws to anchor the plates, and then we'll use 2 screws that go across the fracture plane. That's critical because we've actually hired, what are they? A third party that will be able to verify on radiology that we have healing. It's really more of a healing study than it is anything else.
Got it. It's fusion. It's fusion versus some control. Yeah.
Yeah. We want to basically, and it's most patients the dilemma here we have is most ankle fracture patients are gone at 6 months. They're pretty much out of the system. They don't want to come back to their doctors. And so we don't want to have an extreme fallout, even though the FDA wants us to follow them for 9 months. So we're going back and forth right now on how do we not have a huge fallout and never complete the study where, and it's because it's the system now more so than anything else.
Yep.
You're out.
Patient
So,
selection's
so,
so
um,
hard in studies.
so patient selection is going to be critical. We're going to also include where the study is approved for 10 centers. Um, we'll probably have a blend now where, 4 or 5 years ago, we probably would have gone with 90% hospital-based procedures. Now it's probably going to be 50% ASC and 50% hospital. And the reason for it is a lot of the patients are done in ambulatory surgical clinics now.
It makes sense. That makes a lot of sense. Are there any, can you tell us about the manufacturing part of it? I mean, behind the scenes can you scale, from 1,000 to 10,000 to 100,000
Yes, we
implants?
have we have
Okay.
all the capability of scaling both for screw manufacture that we have. We have dealt with two in the past. One is extremely high quality. We actually have to, when you perform a 510(k), you actually pick the number one product on the market. And you still have to do the three standard tests of pull out force, 3-point bend, and torque diffusion. So if you're doing all three, so we have to code our product and then go not just do biocompatibility, but we actually have to demonstrate against a predicate, the predicate being a standard, well-known product that's all in the market that has a K number. And we have to be able to, so you can't predicate against yourself. Um, and we have to show that we don't affect the torque to failure
Yeah, that
or
should be easy. That
or
should be
three
easy.
point bend.
You're not changing the dimensions of the screw, so that's kind of a no-brainer. Yeah, those mechanical
And
tests.
but you picked up another one that I've never didn't mention. Then we do all of our microbial tests using 4 millimeter by 10 millimeter screws. We don't use titanium discs. We actually
Hmm.
use the screw itself. And the reason for it is the screw itself has, nooks and crannies and everything else along the, um, you know, along the line. So you have to be careful to make sure that, again, we're testing apples to apples, and that is we're not putting a disc or a 2.5
Yeah,
millimeter, 5 millimeter thick titanium disc into the body. We're putting a screw into the body or a plate. The plate's less of a concern, believe it or not, because the plate sits on the periosteum of the bone, so it sits in soft tissue for the most part. So,
hmm,
so the biggest concern are bone screws because bone screws perforate the cortical edge, and they're the ones that contaminate the cancellus or the inner portion of the bone.
yeah.
So the
Create the
bone plate is
pathways.
not the
Yeah,
major concern, it's the screw.
interesting. All right, Mark's back. Rish, Andrea, anybody have a question before we wrap up? Go ahead, Mark.
So I had to leave earlier, and you were talking about plates and screws. Are you doing anything with total joints? And you probably are. I just missed it.
We've been able to coat total joints, but we're going to be looking for a partner in this area, and that
Yeah.
is because we're not going to develop a total joint. So,
Okay.
you take Zimmer. I mean, Zimmer owns what, 25 to 30% of the hip and knee total joint space. So you take them as an example. It's kind of hard to go off and develop your own or partner with a
Yeah.
small company.
Well, I guess really all I meant was you could coat one.
Oh, absolutely.
Okay, that was really what I was talking about.
Yeah, not a
And
problem.
one of the
Yeah,
people on
Mark,
the
they
go
can
ahead.
coat anything. They can even coat PEEK. I mean, they can coat any metal. It's incredible.
Cool. Now,
Rish, am I pronounced your name right?
Hey, Tiger. It's Rishi, but no worries, no
Okay,
worries.
Rish. All right, Rish.
Hey, Paul. Thanks for the presentation today. Just quickly, did you have to test if the coating corrosion affected the strength of the screw at all?
Yes, we actually coated the screws, and that's where we had to perform the standard 3 test of the
Okay,
torque to failure, 3-point bend, and torque to failure, 3-point bend, and
Pull out is the
pull
third
out
one. Yeah.
force. Yeah,
okay,
and we are unaffected.
okay.
In fact, what we have found is um, platinum when we bond the way we bond our sputter silver and platinum onto our device, we actually had to do abrasion testing too, where you send your screw, this is where it's against a wheel and it spins and it looks at it versus titanium by itself. And our platinum actually provides strength over standard titanium
Hmm.
in an abrasion testing.
Okay.
Interesting.
Yep.
Yeah, this stuff's only angstroms thick, but it changes. It's amazing how it changes it makes it antimicrobial without really changing. Go ahead, Mark.
so Paul, you're probably up on this, but is there a big difference in surgical site infections between a freestanding ASC versus a hospital inpatient surgery center or hospital main OR? And because I did a brief look, through chat and Claude on those numbers, and the cost is horrific. I,
So the cost, so if you have an infection from an ASC, it's going to be tremendous. It's going to be much more costly. Why? Because you can't keep the patient overnight. You're going to have to transfer them to the hospital. And so that's going to drive the cost up significantly. Whereas if you have a patient who's already in the hospital, that's a complication they can deal with there. I haven't seen any data yet that's come out and said that infection rates are higher or lower in ASCs versus hospitals. I would suspect over time that because there's so much variability from one ASC to the next ASC, that you will see something related to that. But if there was a study done, oh gosh, about 6 or 7 years ago, and it was published in JAMA, and it showed in neurosurgical procedures, spine procedures, that the longer the procedure, the more contamination from
oh yeah,
the ambient air. And so at the beginning, they would swab everything that sat on the circulating table in the back, and they would swab all that for contamination at the beginning of the procedure. And then if you had a procedure that was like 6 hours long, they would swab that. And the contamination rate went from zero, literally zero, to 15 to 20%. So as the procedure becomes longer, so where would we fit quite well? In a long neurosurgical spine procedure. And one of our co-founders,
Revisions too. Yeah.
a revision procedure. One of our co-founders is Dr. Bowden Chopko, who's assistant chief at Ohio Health, and he specializes in cranial-based neurosurgery as well as complex spine. But he does these scoliosis redo patients. He gets a nursing shift in the middle of his procedures because he's sitting there for so long. So you can
yeah.
imagine the contamination rate on those type of procedures.
So Tiger, one of the guys on the call here is Daniel Slatter. He's an orthopedic surgeon who put a question in the
chat.
You put a question in the QA, question and answer.
Got it.
Do you
I
see
didn't
it?
see it.
Yeah.
I will.
Maybe he could put it in the chat.
All right, here we go. I'm going to ask Dr. Slatter's question. Great technology. If O2 and water are required for the metal reaction, what does the partial pressure of O2 need to be, and should it be a wound, be left open briefly to ignite the process? Oh, really? What a great specific question.
That's a great specific question, and that one I don't know.
Well, we'll get you an answer.
Schneider has he has
You
a
know,
special
when
interest
I'll check with
this.
my, my vice president of R&D, Dr. Houdin Danan, who actually is an electrochemist. He did his electrochemist PhD from developing
Houdan's
silver
got his
as an
hand. Yeah, Houdan's got his
anti-germicidal.
hand raised. So we'll yeah, jump in, Houdan.
Houdin, you're on.
Yes.
Houdin.
Can you hear me?
Yes.
Okay. All the corrosion reaction you see, iron, whatever you see, the principle of the reaction is water and O2 going to OH negative and taking the electron from the metal. Any metal you see corroding, that's the reaction. Amount of the oxygen needed is extremely low. So if to prevent the corrosion from happening, basically you cannot have much of an oxygen, and that would damage the tissue, and the problems with lack of oxygen will show up to other things rather than the system not working. In fact, in real human body, that never happens. Unless in really, really bad cases that I have never seen from. Theoretically, yes. But you cannot have any tissue or any bacteria or whatever grows unless they're anaerobic that grow in a place that doesn't have any oxygen.
Well, there's the answer. Hopefully that satisfied Dr. Slatter.
Very good. Thank you, Houdin.
Thank you, Dean. So we're kind of hit the top of the hour. Fascinating. I mean, if I were a patient, man, I know what I would want. So we're pulling for you to get on the market in the US.
Well, I just found out that I may be a patient last week that I may need a partial knee, and I'm not too excited about it. But I've been thinking, gosh, if I need a partial knee, am I going to go get one and get it coded so I can get I'll be the first knee implant patient.
Cool. Do the tibial side and the tibial screws. Yeah, for sure.
There you go.
All right. Well, thanks, Paul. This has been great. This is we're pulling for you.
Thank you very much.
Yeah. Thanks.
I enjoyed it. Thank you again, Tiger.
Thank you.
Good luck.
Bye now.